Intriguing and informative. And so if everyone could come in and take your seats. Thank you so much. Mucho gusto.
Okay, I'm going to go ahead and get started. First of all, I want to thank the organizers for including me in this groundbreaking event. It's really honoring to be here with such an esteemed group of speakers. I've learned so much already, and I think it's great that everybody comes together to all learn together. My name is Hudalla. I'm the founder and chief scientific officer of ProVerde Laboratories. We are an analytical testing facility that is focused on cannabis and hemp analytics. We were founded back in 2013. Among the first ISO 78025 accredited laboratories for doing cannabis testing in the United States. We have, from the beginning of our company, been founded on the principle of using high technology for analyzing cannabis with cutting-edge science and instrumentation. And with that, we work with cultivators, producers, patients, researchers, and clinicians. About the plant, cannabis sativa, we've heard a lot about that here already. Our history with cannabis goes back over 7,000 years. With regulation in the United States, only within the last 100 years, and became a Schedule I compound in 1970. When it was scheduled as a Schedule I compound, this resulted in significant impediments to science and research, medical treatments, therapeutics, and it was a shame that we couldn't do the research on such a miraculous plant. Even with that prohibition, there's been significant evidence presented to support the medical use of cannabis for a number of diseases and conditions. In the United States currently, there are 36 states that have some form of medical marijuana programs in place, with 18 states and the District of Columbia which permit adult use, treating cannabis very similar to alcohol. I have these numbers in red, 36 and 18, because they're constantly changing. And it's my reminder every time I give a talk that I have to update these. It changes daily sometimes. Then with the recent passage of the US Farm Bill, they've had significant impact on the cultivation and the production of hemp products. A lot of the cannabis that I talked about today is based on hemp or cannabis that is grown primarily for cannabinoid production. Right now, our laboratory does not currently focus on hemp for fiber testing yet. If we look at medical cannabis programs around the world, this is another slide that I have to update frequently. There's at least 59 countries which have some level of medical cannabis programs, and other countries which have programs that are specific to hemp only with no allowance for the marijuana type strains. When I think back about cannabis and its history of testing, going back 10 years ago, they were testing cannabis for cannabinoids, but the analytical data was used as a marketing tool to show how much THC was in your cannabis strain. My strain has more than your strain. Because it was used as a marketing tool, the data that was collected and promoted was sometimes suspect. It was manipulated for financial gain because it wasn't about consumer safety. It was about getting a higher price for your cannabis. And so anybody with a small GC, gas chromatograph system, a garage, and an analytical balance could therefore become a cannabis testing lab. Things are more complicated now, though. This is a new era for the cannabis industry. We see large-scale investment in these cannabis operations in the millions of dollars, large-scale production for cultivation and processing. And so as we transition to this larger scale manufacturing process, we have to start looking at GMP operations. Today, I still explain to some producers that GMP does not mean good marijuana product. And so we have to start looking at the whole process, the raw materials, optimizing the manufacturing processes, that could be extractions, formulations, and most importantly, monitoring quality control of the products that are being produced that are typically going into consumer consumption. And so the laboratories like ProVerde Laboratories that are providing these services are becoming more regulated. We're responsible to state auditors. We are typically governed under ISO 17025 analytical testing standard. There's very stringent limits set for the contaminants that we are testing for. And because of that, the analytical laboratory itself requires a significant capital investment in terms of the instrumentation that's required, the staff, the level of staffing that's required, and it's become very challenging. As I look at the role of analytical chemistry in cannabis, I can pick out different points of the industry where we play a critical role. In the earliest stages of cultivation, we can use our analytical technologies to help select desirable chemotypes or the chemical expression of strains. Even when they're 2 inches tall, I can tell what the cannabinoid production is going to be like, which types of cannabinoids. And so we can also use our analytical instrumentation and methods to optimize cultivation, looking at nutrient uptake and things like that. After the plant is harvested, then it goes typically into extraction oftentimes, and so we can use analytical technologies to optimize these extraction processes and to determine the profile and potency of the resulting extracts. In the production realm, we're very active with producers in the research and development of new formulations, novel delivery systems, and always at the end of the day, we have to provide the information for labeling, for understanding the profile, the cannabinoid profile and the potency. We also do a lot with shelf life testing. It's one thing to produce a product, but you also have to document and demonstrate that you have adequate shelf life or stability of your product. And then for QC and labeling, again, we're going to look at the profile and the potency of the cannabinoids, the terpenes. We also are focused at looking on meeting the regulatory requirements to ensure that products that are going into the consumer market are free from contaminants that could be potentially harmful to consumers. All of it is focused at the end of the day in ensuring consumer safety. So as I look at the cannabis supply chain, you have the cultivation, the farmers or indoor cultivation. Post-harvest, there's drying, curing, trimming activities that go on. There's extraction processes and formulation processes, which all result, at the end of the day, in a product that is ready for consumer consumption. But as I look at this cannabis supply chain as an analytical chemist responsible for consumer safety, I'm going to look at the same cannabis supply chain as a contaminant supply chain. Where do contaminants enter the process? Why am I concerned about that? Why are regulators concerned about that? What do consumers need to be wary of? So when we talk about cultivation, heavy metals can enter the stream from contaminated soil, water, fertilizers, or nutrients. Pests, insects, and rodents can be problematic. Certainly microbial contaminants is a big concern, as cannabis is oftentimes subject to yeast and mold contamination. It can be very challenging. Mycotoxins is another contaminant. It's actually I call it mold poop, but it's a secondary excretion of certain mold or fungi. And then pesticides. We as humans, trying to mitigate all of these other pests and microbial contaminants, are oftentimes using pesticides to control these. And so human interaction plays a role in the contaminants for that. As I look at the drying, curing, and trimming, how the product is dried, the conditions, the temperature, humidity, those can all play a role in potential microbial contamination being propagated. Equipment used for trimming can be contaminated. If you're using cleaning solvents on that trimming equipment and then you go to trim another plant, you can cross-contaminate with solvents or cleaning solutions. Microbial contaminants can also crop up. If your plant material has mold spores on it and it's dried too slowly, that mold can propagate. The people doing the work, handling the product, can also, through maybe poor hygiene or inadvertent contamination, can contaminate product as well. For extraction, most extraction equipment, whether hydrocarbon or ethanol, are made out of some form of metal, stainless steel, and those metals can be introduced into the extraction stream and contaminate the product. Residual solvents, it's fine to use solvents to extract the cannabinoids from the plant, but before it goes into the consumer stream, you want to make sure that those solvents are removed. Pesticides. If the plant material has pesticides on it, when you concentrate the cannabinoids through an extraction process, you're also potentially concentrating the pesticides. And again, as you'll see in every case, handling, human interaction and hygiene is a critical role that has to be considered with the introduction of any contaminant. When that extract is then formulated, oftentimes other ingredients are added, oils, flavorings, other excipients. Those themselves can contain contaminants which are then introduced into the product. Packaging materials is something that the cannabis industry is fairly new to thinking about. In the pharmaceutical industry, we've been thinking about this for decades. But it's kind of a new concept to the cannabis industry that the packaging material that we put our products in can actually impart contaminants into our products. As you saw at every step of the way, human interaction and hygiene is one of the most concerning roots of contamination. As an analytical chemist, I have a number of analytical tools in my toolbox that I can use to test for these different contaminants. Which tool to use depends on the level of sensitivity I need, what I'm trying to test for. At the end of the day, each one of these tools is very expensive, and so that leads to the capital investment required to set up a testing laboratory. As an analytical chemist, one of my primary objectives is to take things apart, to see what's in them, what components are in them. We all know that cannabis has a number of cannabinoids and a number of terpenes. That's why we love this plant so much. What everybody may not be aware of is that there's many other chemical compounds that are in there. Hydrocarbons, proteins, amino acids, flavonoids, sugars, even a vitamin. And so cannabis, like other natural products, is a very complex plant, which makes it challenging at times to analyze and to pull all those components apart for a better understanding of the plant. I think most people know about Trichomes. That's the factory in which the cannabinoids and terpenes are manufactured. The cannabinoids manufacture the terpenes, and the terpenes can be found all over the plant, on the stalk, the leaf, but most abundantly in the flower. It's interesting that the trichomes are not found in the seed, and they're not found in the root. Most of extraction activities and consumption activities are focused on the flower because of the high density of trichomes there. So what happens in the trichome? There's a genetic biosynthetic pathway for cannabinoids with CBGA, which I call the granddaddy of all cannabinoids. It's the starting point for the cannabinoids. That is what's produced in the trichome initially. It's through enzymatic reactions that CBGA will be converted into CBDA and THCA. Further degradation of the THCA can form CBNA. We're going to have a whole alphabet soup here that you'll be quizzed on afterwards. But then decarboxylation, removal of that acid group, results in the neutral version of the cannabinoids, which are not inherently produced by the plant. The plant produces the acid form. It's only through heat or decarboxylation that the neutral form is created. Everybody knows about THC. That's because it is the most psychoactive of all the cannabinoids found in the cannabis plant. And so oftentimes the neutral form of the cannabinoid is referred to the active form of cannabinoids. But from a bioavailability, biotherapeutic standpoint, all of the cannabinoids have some level of activity and are equally important. If I look at some of the non-psychoactive cannabinoids, these are all neutral cannabinoids. This is a paper that was published in 2009 that looked at all the different pathways within the human body where the cannabinoids might be involved in some therapeutic potential. And so looking at cannabis as a medicinal product, there's a lot of cannabinoids that we can see as tools that can be used for a therapeutic perspective. How do we analyze most of the things that we're looking at? We use a technique called chromatography. That can be gas chromatography or liquid chromatography. Essentially, they're very similar in that we're going to take parts of the plant and we're going to pass it through a long column. We're going to push those through the column either with gas and temperature or with liquid. What we record on the other end of the column, we call a chromatogram, but it's a series of peaks. These peaks can give us information about the nature of that chemical compound that was found in that plant. And we can look at the time or the retention time to help identify the nature of that chemical compound. Those chemical compounds can be good. They could be cannabinoids. They could be indicative of the presence of terpenes, vitamins, other chemical compounds. But they can also indicate the presence of contaminants. It might be pesticides, could be heavy metals. There's many different forms of cannabinoids that, depending on the technology that we use, will show up in our chromatography or analytical results. Once we understand which chemical compound I'm looking at, I can then look at the size of the peak or the area of the peak to understand how much is there. And this is how we're going to analyze a product to see how much CBD. Does it meet the amount that's on the label? We would also use the same to identify how much pesticide is there. Is this above the threshold for safety limits? This is just an example of certified reference standards. These are of known concentration. We mix them all together. We analyze these routinely to make sure that our instrumentation is working well. And you can see that we have very good separation and identification of multiple cannabinoids. So what do we do with this tool? As a cannabis lab, I would say that we see some of the best in terms of plant product, consumer products, but I also see the worst of the worst. And so I'm going to show you some examples of good things and bad things and how we use this data to help producers create better products. When we are working with cultivators, very oftentimes they will start growing multiple seeds. And without waiting till the plant is fully mature, we can start getting an idea with the very first set of leaves, which cannabinoids are going to be expressed. Is this going to be a THC dominant strain? Is it going to be a CBD dominant strain? In this particular experiment, we collected a leaf, each leaf that came out, the first leaf, the second leaf, the third leaf. We followed it all the way to maturity. One of the things that we saw is that the expression of individual cannabinoids, or the profile, did not change through the life cycle of the plant. What did change is how much was there. So these peaks got bigger and bigger and bigger as the plant got bigger, produced eventually the flowers and more trichomes. And so we use this technology to help cultivators decide which seeds they want to move forward with. It takes a long time. It's expensive to pop 100 seeds, and you don't want to move all those forward to maturity to decide which ones you want to move forward with. So we can tell very early on what it's going to look like. We can also help understand chemical processes. This is a typical chromatogram for a dry cured flower bud. It's predominantly THCA, which is the acid form of THC. This is what a normal marijuana strain would look like. But if the strain or that particular flower sample ages, if it gets old, we would expect, based on the pathways I showed earlier, I would expect a certain amount of degradation of that THCA forming THC or CBNA. both of which can further degrade to CBN. So this particular sample, after 5 years, you can see that the THCA, which was the primary cannabinoid when it was fresh, is almost completely degraded. And you can see that there's a significant contribution from CBN, which is the oxidative degradation of THC. We also help producers optimize their processes. This is an example of a patient who was trying to create a tincture, and they were trying to create a neutral, a THC, a psychoactive tincture. And you can see that with certain heating, they did not have a complete decarboxylation. What they were going for was a complete conversion of the THCA to THC. With a little instruction, using a little bit higher temperatures, a little bit longer time for the decarboxylation. This is the same sample when he brought it back 2 weeks later, and he had completed the effect all the way from the acid had all been converted to his target THC, which is what he was interested in. And so that got me to thinking, how fast does decarboxylation happen? Does it happen faster at higher temperatures? What are the kinetics? And so we started studying this as a function of temperature. We started following the chromatography over time at different temperatures. And with each time point, you can see that the CBDA signal, this is a hemp strain, so the CBDA is decreasing while the CBD is increasing. You're seeing the conversion of the CBDA to the CBD. We performed that experiment at multiple temperatures, and so we can start plotting as a function of time and temperature what those rates of conversion look like. And so if you're a producer and you want to go all the way to a fully decarboxylated product, understanding the kinetics or the rate of this decarboxylation can help you optimize your processes. Going back 10, 15 years, I would say that the majority of the cannabis was probably smoked. These days, we see derivative products being made into almost every form of consumable product. In order to get a flower into a consumable product like that, typically involves some form of extraction. We see ethanol extractions being very common. Ethanol has some advantages. It's food grade, fairly non-toxic. It can be recovered after the extraction and reused. So there's a lot of benefits to it. Hydrocarbons are also used extensively in the United States, oftentimes with butanes or propane. There's advantages and disadvantages to all of these. One of the disadvantages of hydrocarbon extractions is it's highly flammable and it can be dangerous. It oftentimes requires special facility considerations to make sure that you're doing this in an explosion-proof room. We also see a lot of extractors using supercritical fluid, carbon dioxide. CO2 is a non-toxic, non-flammable gas, which when pressurized becomes a liquid. And that liquid, as a pressurized gas, can be used to extract the cannabinoids and terpenes out of the plant material. And if it's done appropriately, it can be done such that it maintains a very terpene-rich extract. Whichever extraction process is used, there's typically some level of post-extraction processing to remove solvents, to remove fats and waxes, but that final extract then can be used as the basis for many products. And so some of the derivative products that we see would look like this: extracts and concentrates, tinctures, ready-dose infusion that are alcohol-based or glycerol-based. Topical salves and lotions are very therapeutic. They work excellent for certain conditions like arthritis. Edibles. Edibles are oftentimes a delivery of choice because they're very easy. They're tasty sometimes. I have a hard time not eating these brownies in the lab, but they are a good dosing mechanism. And then vape pens have become very common. Which product a consumer might use is going to depend on a lot of things. What kind of conditions are they using it for? What kind of relief are they seeking? And then again, they all have different advantages and disadvantages. One of the disadvantages for edibles is the time to onset for that feeling of relief or the psychoactivity to kick in can oftentimes be 90 to 120 minutes. Whereas when consuming a vape pen or smoking marijuana, it's a much faster response. And so when I'm thinking about all these products, I start thinking about nature versus human nature. How do we cultivate? How do we process the plant? And how do we do it without introducing contaminants? How do we measure the contaminants? Because there's many tools in our toolbox. It depends on what the regulatory requirements are for how low we have to go in our measurement. And then how do we regulate these contaminants? One of the challenges with regulation of contaminants is there's very little known, partly because of the prohibition of cannabis, there's very little known about how the contaminants in cannabis interact with the human body. Certainly pesticides have been studied for years, but they've been studied in an agricultural sense. We understand if your fruits or vegetables are contaminated with pesticides, they go into your digestive tract. We understand some of the toxicity of that. But there's very little data on what happens to these pesticides when inhaled, or even worse, when they're heated, and that heat degrades the pesticide, and now you're inhaling those degradations. So there's a number of considerations when you're talking about some of these limits. And so when you think about appropriate limits of contamination, I like to think about what is the route of administration. Whether it's going to be inhaled, I might be particularly concerned about pesticides or mold spores. If it's basically being ingested, I might be very concerned about heavy metals and pesticides. With topicals, I have different considerations or different concerns. And so how a product is to be used should be taken into consideration when we think about some of the different contaminants. And so many of the contaminants that we focus on are pesticides, plant growth regulators, heavy metals, the microbial contaminants are always present or can be, and the processing solvents. And so I'll show you some examples of all these. Oftentimes I take a lot of the input for my talks directly from the news. We see it all the time, headlines. This is one where in Colorado, the Department of Agriculture found numerous pesticides in some product and it resulted in a recall. One of the challenges with pesticide analysis is there's many different pesticides. In the United States, the requirements for testing pesticides varies dramatically from one state to the next, both in terms of which pesticides are permitted for use and which ones are required for testing. Ultimately, we need to ensure that the pesticides aren't there, that the products that are going into the consumer market are free from these pesticides. And again, as I mentioned, there's little toxicity information available for these pesticides with regard to inhalation processes. It gets even more complicated. As I said previously, if you have flower material that is contaminated with pesticides, that oftentimes go into an extraction process because you're trying to concentrate the THC or concentrate the CBD. This is an example of a pesticide result. The pesticide has microbutanol in a flower sample. But when that flower sample was extracted and we tested the pesticides, we saw a similar increase in concentration. Just like we would see an increase in THC concentration or CBD, the pesticides are increased as well. That has multiple concerns for me. Number one, you've created a very contaminated product now. But another consideration is now the equipment that I've used for my extraction is now contaminated. And it's pretty likely that the next batch of process of flower going through that extraction, even if it's clean from all pesticides, is now going to have pesticide contamination left over from this extraction. So that's something that people have to be considering. And even still, as much as we try and prevent these, we still see products in the market that are contaminated with pesticides. This is a vape pen. vape cartridge. And these are the peaks or the signals which are indicative of the pesticides that are present in that sample. And if you look at some of the levels, 296,000 for bifenthrin. In Massachusetts, the limit is 10. So it's a little bit over that. This particular producer had the audacity to label their packaging with a big sign that says pesticide free. While it's not very funny, we're bored in the lab, we joke about it being free pesticides, which would be more appropriately labeled. Another thing that we have to consider is not just the pesticide in the vape pen or in the product. This is another example of a vape cartridge that had myclobutanol. Myclobutanol is fairly non-toxic as a pesticide when ingested. Don't do this, but you can probably drink it without huge concern. The problem is when you heat it, if you're smoking marijuana that has myclobutanol on it, the heat of smoking it or combustion degrades the myclobutanol pesticide into multiple degradation products, one of which is cyanide. You do not want to be inhaling cyanide. Not good for you. Heavy metal toxicity is also something that needs to be considered. We know that cannabis is very adept at bioaccumulating toxic heavy metals from the soil. I think it was Dr. Uma yesterday who was talking about remediation of soil for different heavy metals and radioactive compounds. Again, most of the toxicity data is in reference to environmental exposure of heavy metals. But we can also see the heavy metals in the mainstream smoke off of vape pens and flower when it's being smoked. And so again, there's more toxicological research which is needed to understand the implications of these contaminants. So at this point, it's very challenging for regulators to set a safety level. They're setting a safety level with very limited data, and that's a dangerous position to be. But we see heavy metal can be introduced from the water, it can be introduced in the soil or growing medium, or from the nutrients used for cultivation. We've done some examples where we have shown that extraction is a viable route for remediation. So if you have plant material that's contaminated with heavy metals and you extract it, the resulting extract will have very little heavy metal contamination. The reason is that the metals are not very soluble in the liquids that we're using to pull the cannabinoids up. And so the heavy metals are left behind in the plant material. Here's an example of metal contamination from the extraction equipment itself. So this was a brand new piece of equipment. Oftentimes the stainless steel fittings, they're all threaded. And in this particular case, there were leftover shards of metal that were left in the flow path. And so when cannabis was extracted, that flow path of cannabis extraction picked up these metals and could be seen in the raw material. We also look at vape hardware. So it's not always just the cannabis. It's not always just the cannabis extracts. Sometimes it's the containers or the hardware that we use. In this particular case, we collected the vapor on basically a cigarette filter. We were analyzing these cigarette filters for metals. What we recorded was aluminum, copper, nickel, and chromium. These are metals you wouldn't typically find in high levels in cannabis, but you do find them in heater coils that are in vape pens. One of the big challenges for us in the United States is almost all of our vape pen hardware comes out of China. And whether you're buying high quality vape hardware or low quality vape hardware, they look the same. It's very difficult to distinguish. There was also, not too long ago, a recall from the FDA on CBD tinctures that were contaminated with lead. This is not uncommon for us. And then oftentimes we will, from a producer, get the bulk ingredients for testing and show that there's no heavy metals in the tincture. When they go ahead and bottle it and submit it to us for testing, all of a sudden I see heavy metals that were not there before. If I test that same sample 2 weeks later, that amount has gone up dramatically. This is 44,000 parts per billion. The acceptable level under the United States Pharmacopoeia is 1,000 parts per billion. If I test that again 2 weeks later, the amount is going up. And so this is a case where the bottle itself is contaminating the product. And what we did through exhaustive research studies is we showed that the bottle itself, the packaging, was contaminating the material. And after studying it, realized that it was coming from bottles that were manufactured with recycled glass. Residual solvents, we think of solvents being used for extraction, and those are ones that we have to test for. But solvents are also used in cleaning equipment between processes. And so if that equipment is cleaned with a solvent and it's not treated appropriately, that cleaning solvent or cleaning solution can then end up in your next batch that is extracted. So those are other concerns. This is just an example of butane. These are just a couple of examples. E-liquid, this is the extracts or concentrates that are going in vape pens. We see a lot of problems with pesticides, different solvents, additives, like polyethylene glycol, propylene glycol. Vitamin E acetate is particularly concerning, and flavorings. In the US over the last couple of years, we had quite an issue with something that has now been coined E-Valley. It was an e-cigarette or vaping-associated lung injury. It was given that acronym by the CDC, the Centers for Disease Control. And they did so because they found at least 2,800 cases of E-Valley that resulted in hospitalization of consumers, the youngest of which was 15 years old. 68 of these cases resulted in fatality. They died because they smoked a vape pen. The E-Valley was thought to be caused from the addition of vitamin E acetate as a carrier to these cannabis oils. Certainly there could be other contaminants of concern, but the vitamin E acetate is one that's critically important. When I look at the chromatography, this is what I see that's concerning. You can see that there's a very large vitamin E acetate peak and a very small delta 9 peak. You don't want to see that vitamin E acetate peak, it shouldn't be there. This is just another example of a vape cartridge with another additive, polyethylene glycol. Oftentimes these additives are not well studied on how they fare in the lung. What do they do when they interact with your lung tissue? CBD isolates have become very common over the last couple of years. And typically, CBD isolates are recrystallized in an organic solvent, typically heptane or pentane. They used to use hexane, but because of toxicity, they moved apart from that. With this particular example, we saw a lot of solvents that are particularly concerning. They're known carcinogens. And so when I contacted the producer and said, what are you using to produce your CBD isolate? He sent me a picture of what he used to produce his product, which is going into formulations for consumers. It was a can of paint remover, paint stripper. It works great to make a toxic product. Don't do it. We also use a lot of our tools for research. When we look at some of these concentrates, oftentimes we report that it's 80% purity, 88% cannabinoid. The question is, what's the remaining 12%? As I said before, the complexity of the cannabis plant is amazing. Sometimes we can see hundreds of chemical signatures or peaks that relate to the cannabinoids, the terpenes, the phospholipids, all these other chemical compounds that are in the plant. We see those. Microbiological contaminants I talked about, many of the extraction processes actually kill the microbes. So extraction is kind of a means of remediation. More recently, we see a lot of therapies that involve the acidic form of the cannabinoids. In order to maintain the acid in the acid form, we can't heat those products. And so normally the sterilization that happens at the high temperatures is not happening, and so those are a particular concern. This comes out of the news yesterday. In California, there was a recall for cannabis flower that was contaminated with black mold. Black mold is something you do not want in your lungs. Think of a mold spore that gets into your lungs. The inside of your lungs is dark, moist, warm, perfect breeding ground for mold. Like most of our other analytical techniques, we have multiple tools in our box, whether we're culturing, we're looking at DNA. There's a variety of techniques depending on what information we're looking for. At the end of the day, all of our results are put into a certificate of analysis. Certificate of analysis is a unique record documenting the testing that we performed. All the data on that is traceable to the NIST standards. NIST is the National Institute of Standards and Technologies in the United States. We include a QR code or a 2D barcode that can be used to link to the original certificate, which is secured in a cloud-based database to prevent people from manipulating the data. It discourages alterations and counterfeit. But is that secure enough? There's organizations, CanID is one of them, that is looking at the next level of security by looking at blockchain technology to protect this data from being manipulated. That's the future is we need to be more secure with our documentation. ISO accreditation, I'll just talk briefly about that. This is the accrediting body under which our laboratory is certified to make sure that our laboratory performs good laboratory practices. It provides the states, the regulators, and consumers a level of safety, understanding that we know what we're doing. We have some competency. We have to demonstrate that competency through proficiency testing, calibrating our instruments, validating our methods, an incredible amount of documentation. We have standard operating procedures in our facility that tell us how to write a standard operating procedure. Other considerations that are important is the country, state, local, and regulatory agencies. Communication with them is key because once regulations are put in place, they're very difficult to change, so you need strong communication. The accrediting bodies, who is going to certify a laboratory or an organization? Instrument or consumable vendors that we buy our equipment from, our chemical supplies, making sure that they have quality components. There's many organizations that have been established to help establish standards for the cannabis industry so that regulators can have another degree of confidence. The United States Pharmacopeia, AOAC, ASTM. Again, it's more alphabet soup. These are all professional organizations that have a history of regulatory compliance. We need more collaboration between cannabis testing laboratories. Standardized testing methodologies would help, and again, the proficiency testing. When we do a proficiency test, we pay money to have somebody send us an unknown sample, and we report those results back, and we get graded to see how we did. And those are results that we can then give to our regulators so that they can have confidence that we know what we're doing. So just to summarize and wrap up here, chemical understanding of cannabis is essential for the rapidly growing global economy with cannabis and hemp. Contaminants, the toxicity is often cumulative, so for heavy metals, pesticides, oftentimes the people we treat are all the way from several months old to 98 years old. And so if you look at the most fragile population, infants or senior citizens, those contaminants are really of huge concern. Our primary goal is not to pass or fail the producer's product. Our primary goal is to ensure consumer safety. If we find contaminants in a product, we're a fantastic resource for helping producers figure out how to either get rid of them or how to prevent it from happening again. We treat our customers or our clients as partners because we're not successful without their business. So we like to always say we're in this together. A lot of this work I didn't talk myself. We have an awesome laboratory team in Massachusetts, which is getting 3 feet of snow tomorrow, so I'm also very happy to be here for that. But more than that, I would like to thank you for attending my talk. Thanks. If there's any questions, I'd be happy to